Hi, and welcome to Chapter 14. Today we're going to learn all about gene expression, which is basically how you go from DNA to making a gene product, usually considered to be something like a protein. We'll get into more about that later, though. So let's start out with part one. And part one is there's a lot of important background information that we're going to go through. So some history, that kind of thing, like how things were discovered the way they were. So let's go ahead and check out some of the background information on gene expression. So to start out with, there is information in DNA, right? We've already talked about how DNA is made up of A's, T's, C's, and G's, and a specific sequence of those nucleotides in DNA actually contains information. We also know that DNA is inherited, like we've been talking about that for a couple chapters too, right? It's inherited and it leads to specific traits, but it does that by basically making proteins. And it turns out that RNA molecules are also involved in the making of proteins, also called protein synthesis, because synthesis just means to make. So proteins are basically the link between the genotype and the phenotype that we've been talking about for a couple chapters now. Gene expression is the process by which DNA makes or directs protein synthesis, and it has two stages, transcription and translation. We're going to go into a lot of details on transcription and translation in this lecture eventually, but not to start out with. So genes do make proteins. Like that's one of the things that we're going to talk about. And again, we'll modify that a little bit at the end after you get to know everything. But first, let's talk about how this relationship was determined. Like how was it figured out that genes, which are found in DNA, and how they make proteins, how was that discovered? So again, some history here. In 1902, we've got this British physician. His name is Archibald Jared, and he's the one who first said that, maybe genes make the phenotype through enzymes that catalyze specific chemical reactions. Now remember that so far, as far as we know, and again, I might modify this a bit later, enzymes are proteins, or they're a type of protein. For the most part, that's true, at least for the beginning part of this. So Dr. Jared thought that symptoms of an inherited disease reflect the ability or inability to make a certain enzyme. So if you've got a disease, what you've got probably is a faulty enzyme. And that cells make and break down molecules in a series of steps called a metabolic pathway. Now, we've talked about metabolic pathways before too. So, and that was way back at the beginning of the semester, but metabolic pathways are a series of steps in which you change one molecule into another, either breaking it down or building it up. So that was the first thing. And then along come these two new researchers, Beadle and Tatum. And what they did was they looked at a mold found on bread called Neurospora crassa. And they thought it would be really easy to detect mutations in this Neurospora, this mold. And so they looked at that. And one of the things that they studied, one of the things that they were most interested in, is the mutations that allowed the fungus to grow on something called minimal medium. So minimal medium is a very specific term. If you ever take any further classes in biology, like microbiology, you will learn all about minimal medium. Okay, that's just what that is. It's just an amount of food that they get. And it's a very, very, very minimal amount of food. It's like not very nutritious. So what they did was they caused some mutations to occur by exposing them to X-rays, right? They exposed the neurospora to X-rays, and that caused mutations. And then they grew those neurospora in minimal medium, but they also grew them in other media, including like valine. Now, I don't know if you remember what valine is. We don't really talk about it much, but it's a kind of amino acid. And then they grew some in lysine, and they grew some in arginine. Now, again, all of these are types of amino acids. So, so they've got all these mutant, these mutant neurospora, these mutant fungi, and they're growing them up in different environments: minimal medium plus valine, minimal medium plus lysine, minimal medium plus arginine. And then they had a control group where they just grew the non-mutated cells on minimal medium, and they compared to see what happened. One thing that they did find is that in all of the mutants that they created, there was one set of mutants that all absolutely required arginine for growth. So right there, so this one they thought was really interesting, because what was determined was that different versions of these mutants, they blocked a step in which they could make their own arginine, which is the fancy word for that is biosynthesis. This mutation blocked that pathway. So that was some good evidence for this idea of a gene influencing an enzyme. If you wanted to know how the regular neurospora can make arginine, what they do is they have 3 genes involved, and that means that they have 3 enzymes involved. So they take this precursor molecule and they change it into ornithine, sorry about that. And then the second enzyme will change ornithine into citrulline. And then the third enzyme will take citrulline and change it into arginine. So that's the pathway that they use to discover that if you mutate the DNA, then you might get a change in the enzyme. It's important to remember that some proteins are not enzymes, right? So if genes make enzymes, then they're making proteins, right? So instead of thinking about this as one gene, one enzyme, we now have this concept of one gene, one protein. So that was pretty good for a little while, but we also know now that many proteins are composed of several subunits, and those subunits of a protein are called polypeptides. So, and each of those subunits in a protein that's got several polypeptides is going to have its own gene. So what Beadle and Tatum decided was that that it was one gene, one protein, but now we say one gene, one polypeptide. Yes, that's what we say, but remember that we often just call polypeptides, we just call them proteins. So it's a little squishy. It's perfectly fine, I think, to say one gene, one protein. That makes sense. It's also appropriate to say one gene, one polypeptide. Again, many proteins are composed of several polypeptides, so that would mean that you would need several different genes in order to make a single polypeptide or protein. I know that was a little confusing, but make sure you're paying attention to the language there. So the basic principles of transcription and translation is that RNA is the bridge between DNA and proteins. So let's remind ourselves of what is involved with RNA. Remember that it's very, very similar to DNA, right? Except for RNA has ribose sugar instead of deoxyribose. And then, of course, RNA has the base uracil or U, whereas in DNA, they have the base thymine or T. So DNA has A's, T's, C's, and G's. And RNA has A's, U's, C's, and G's. Another major difference between DNA and RNA is that RNA is single-stranded, generally speaking. Not always. There are some exceptions, but they aren't very long living. They don't last very long. And that we're going to go through those two processes of transcription and translation in order to go from DNA to protein. Let's start out with the definition of transcription. And transcription is the process by which we make RNA or synthesize RNA using the information in DNA. Transcribe means to write something down. So write scribe is a copy, or somebody who copies something. So transcription is the process of copying DNA into RNA. And the main type of RNA that we're going to talk about today is messenger RNA, or mRNA. This is probably the most important one when we're thinking about the process of making a protein. So messenger RNA is the main intermediary. It's not the only one, and DNA does make all the other RNAs too, but messenger RNAs are our key friend here. So again, let's just focus on mRNA, and we'll introduce other kinds of RNA later. Translation is the process by which you take that mRNA and you use it to code for your polypeptide. And so mRNA that copied from the DNA, right, it's the second step. We're going to use that mRNA template and make our protein. Now, this all happens at the ribosomes, and we learned all about ribosomes when we were learning about cells, and we're going to really dive into the functionality of them in this chapter. One interesting thing about transcription and translation depends on what kind of cell you're talking about. So in bacteria and other prokaryotes, you don't have a nucleus. And so it turns out that translation, where the ribosome comes along and reads the messenger RNA, that can happen while the messenger RNA is still being made by the DNA. Again, that's because all of that stuff is happening in the cytoplasm. There's no barrier between the DNA and the RNA and the ribosome. In eukaryotes, we really do have a barrier, and that barrier is the nuclear envelope. So we have the DNA in the nucleus, and the DNA in the nucleus will go through transcription. It'll make RNA. We'll talk about this idea of RNA processing in a little while. But since we're just going to jump into transcription and translation for now, transcription is happening in the nucleus and translation is happening in the cytoplasm. So those two things happen in different locations, unlike in bacteria, where it's happening in the same basic location. So in a eukaryote, the mRNA has to be transported out of the nucleus in order for it to be translated. And this happens by when a primary transcript, which is the initial RNA transcript, the initial one, and before any processing happens. And all of that is happening right in this kind of area right here, right? DNA making RNA. And actually this whole line right here of DNA makes RNA and RNA makes protein is known as the central dogma of biology, which means that all cells are governed by the cellular chain of command where DNA makes RNA and RNA makes proteins. So one of the things that we really have to understand is that the flow of information from gene to protein is based on something called a triplet code, and triplet just means 3. So the triplet code is a series of non-overlapping nucleotides, 3 in a row. So a good analogy is to think about them as words. The letters in a word are non-overlapping. So the letters of this word don't overlap the letters of this word. And that's what the triplet code is like. So if we look in the DNA, you could think about there being 3 letters in a row. That might be a triplet code. But that's not exactly correct, because where the triplet comes into play is when it's in the mRNA, the messenger RNA. So those 3 non-overlapping, 1, 2, 3 here, 1, 2, 3 here, 1, 2, 3 here. Each of these is a triplet code. And again, we think about this in translation, because for every 3 nucleotides in a row, we get 1 amino acid. So, and again, the details of how that happens, we're going to go into in a little while. But I'm just introducing to you for now the basics of what we mean when we talk about the triplet code. Again, they're non-overlapping, so that's very important. We wouldn't read a codon as these 3 and then like these 3, right? That would not happen. And then these 3, like that would be overlapping. And so that's definitely not something that we think of when we think of the triplet code. So after we understand that this triplet code are words, then the words are non-overlapping, then we do translate those into a chain of amino acids. So during transcription, 1 of 2 DNA strands, and that's a little strange why the word DNA is broken apart there, but okay, just we're dealing with DNA, right? You have 2 strands. One is called the template strand, and the template strand tells the order of the sequence of the nucleotides in the RNA. So if here's our 2 strands, we have this strand here is called the coding strand, and I'm not going to pay attention to that one right now. I'm going to pay attention to the template strand. The template strand is what's copied. So T in DNA goes with A in RNA. A in DNA goes with U in RNA, because remember, RNA doesn't have T's. C goes with G, T goes with A goes with U, right? So there we go. Now, you might wonder why the other strand is called the coding strand. And that's called the coding strand because it looks so much like the code that the RNA is. So A, and there's a T, but it's a U in RNA, right? G, A, U, C, U. So the coding strand really does look like the mRNA transcript. That's why it's called coding. But it doesn't have anything to do with, it's not copied or anything. The template strand is what's copied. That's our most important one there. The template strand makes the RNA, and then of course the RNA gives information to make the polypeptide. The template strand is always the same strand for any gene. So for you and me, we both make hemoglobin, right? So it's always going to be on the same strand for any given gene. But that doesn't mean that somewhere upstream or downstream from this DNA that the other side isn't the template strand. It could be. It does. There's not like one specific side of the DNA that's the template strand. It's only considered the template strand for the particular gene. Again, it could be the other side for another gene or for another completely different chromosome. So when we look at translation and we look at the concept of the triplets, we use the term codon. This is a very important term. So codons are triplets in the mRNA. And we do read them from 5' to 3'. We've been talking about directionality a bit already, so hopefully that makes sense to you. If not, you might want to go back and look at the information that we talked about for DNA. Each codon specifies or determines an amino acid, and there are only 20 amino acids. And so each codon calls in an amino acid. Again, we'll go into all the details about how that works in a little while, but for now, just kind of remember that. The term codon is used for the DNA nucleotide triplets alongside the non-template strand, or the coding strand is the word I used previously, right? So we sometimes, when we're looking at DNA, we might think about this as a codon, even though, right, because that's the coding strand. So we could think about it that way. But we wouldn't call this stuff down here codons. One other thing that is important to understand is that if you have 4 different nucleotides, right, A, T, C, and G, or if you're talking about RNA, A, U, C, and G, and that you take 3 of those in a row and you get a codon. If you do the math here, what this means is that there could potentially be 64 amino acids, but there aren't. There's only the 20 amino acids. So of those 64 triplets that could be codon, or could be coding for amino acids, 61 of them actually do code for amino acids. 3 of them code for something called stop signals, and so that 61 plus 3 is 64, right? But what about those 20, right? We already know that there's only 20 amino acids. So it turns out that the genetic code is redundant. What that means is that more than one codon can code for the exact same amino acid. We'll take a look at that in a second, but just remember that there should be at least 61 amino acids. There's only 20, so that must mean that some of them are coding for the same thing. The other thing that's really important to remember is that the code is not ambiguous. It's not ambiguous. What this means is that if you've got a codon, it does in fact code for a specific amino acid. It doesn't code for multiple amino acids. It only codes for a specific one. Let's go ahead and take a look at that. So this is a table of codons right here. And if I said to you, let's look up the codon. This is in the messenger RNA, remember. Let's look up the codon that is CCU. So if we do that, the first letter that I said was C. So this is the first letter side. So here's the C. So we know that CCU has to be in this row. The second letter I said was C, right? CCU. So, and let me just write that down for you so you can see what I'm doing. So it's going to be in the intersection of this C and this C. So it's somewhere in this box. And then if you really want to be, very precise about this, you can see that the third letter here is U is the top one. So there it is. Or at this point, I usually just scan around and see if I can find that CCU. So that CCU codes for proline. So does CCC. So does CCA and CCG. All of those codons code for the same amino acid. So that's what we mean when we say that, as I said back up here, that the genetic code is redundant. But it's not ambiguous because anytime you have a CCU, it will code for proline. We also say that codons must be read in the correct reading frame or group. So just kind of keep that in mind, that, again, that's just another way to say that they're not overlapping. They're separate words. So you read one codon at a time, non-overlapping. Finally, kind of to wrap up this part here of the history, it's important to understand that the genetic code is almost completely universal. What this means when I say it's nearly universal is that the same table of codons that we saw here, where CCU codes for proline and ACU codes for threonine. That means that almost everything on this planet, from the simplest bacteria to plants and animals, we all use the same basic table of codon. Table of codons. So this is actually very useful for genetic engineering that we talked about in the last chapter. Genes can be transcribed and translated after being transplanted from one species into another. The only way this makes sense is if this language, this common language of codons, was kind of in operation. It would exist in existence in the ancestor to all living things, and that they passed down this table of codons, this concept of transcription and translation, to all modern day organisms. Well, that's what I have for you for part 1. Let's move on to part 2, where we look at the details of transcription.